Professor George Salmond. I'm an emeritus professor at Cambridge University in the biochemistry department. It's difficult to say how important it is, but it feels really special for me, given the importance of Marjory Stephenson in the history of this society. It's pretty special for me because I teach, or I have taught for many years, actually in the Hopkins building where Marjory Stephenson worked. And in fact, just above her lecture room, we have a Marjory Stephenson lecture room. That's a special thing for me because, you know, I used to go in through those doors every day to teach undergraduates. Well, I wouldn't say, you know, one thing in particular. I mean, because we've worked on so many different things and been funded to work on so many different things. So it's a potpourri of different research activity that's led to the prize, all working with bacteria and some fungi. We work on antibiotics, we work on the ability of bacteriophages to infect bacteria and various other things. So I think it's been a mix of different things. It's not just a single thing because we've done research on things that have interested us, and that's the driver. And of course I've been supported by an enormous number of very talented students, undergraduates, postgraduates, postdocs as well. A particular highlight is the discovery of quorum sensing in bacteria, and we found that in collaboration with groups in Nottingham that we collaborate with. We found that when we were trying to work out how this plant pathogenic bacterium makes an antibiotic, makes a carbapenem, and that led to the discovery of the quorum sensing molecules, and it then became clear that quorum sensing as a phenomenon was very widely distributed amongst bacteria, not just for making antibiotics, but for bioluminescence, for capsule formation, for a whole variety of other phenotypes. And again, that arose because of a purely serendipitous discovery. There wasn't we were interested in how carbapenem antibiotics are made, but we had no idea that all of this was controlled by quorum sensing. Indeed, quorum sensing wasn't actually discovered at that time. It was actually the phenomenon of quorum sensing was originally discovered in bioluminescent bacteria. But then it became abundantly clear that that phenomenon, quorum sensing, is widely distributed amongst bacteria. I think as an academic, you know, a central challenge for every academic is funding, the ability to, you know, keep the show on the road, and that's to get money to pay for students and to pay for postdocs eventually. And that's just a fact of life for an academic. I've been extremely lucky in terms of funding from, you know, major research councils, and I think that's the main thing. Of course, the other thing is, you know, you need to be able to recruit the right people, right? Whether that be students, postgraduate students, undergraduate students, or postdocs. But you need to be able to put all that together. You need the funding. You need the quality of the students. And I've had some tremendous students. I mean, absolutely tremendous. I've at least a couple of these students. I've got a list of them in a slide I'll be putting up. A couple of those students are former winners of the Fleming Prize, both of them. So, and they got the Fleming Prize because they were tremendous students, postgraduate students. I think that's one of the reasons why these prizes are really important, because it gives recognition for, not for the group leader, but for the students who are actually doing the work. And that's really important. vodka. I actually did work on E. coli for some time. I was a postdoc and worked on E. coli cell division with Willie Donachie in Edinburgh. So that was really important because it made you think about a particular thing. And one of my ex-students is here, Jay Hinton, and it's a way of thinking about how you do bacterial genetics, basically. And that means it's applicable to many other systems, which I'm hoping to show. Okay. Okay, so that's my title. You see in the top right here is Marjory Stephenson with her wee dog, I think it was called Judith, and that's right outside the Department of Biochemistry in Cambridge, in Tennis Court Road. The one to the left is a painting by Van Gogh. It's called The Potato Eaters, and I'm going to be saying quite a bit about potato eaters, which is Erwinia or Pectobacterium. There's some potatoes infected in a field up in Dundee. Then down the bottom here, I think I can use this, yeah. Is that working? Yeah. There's an antifungal antibiotic here, and we got into antibiotics some time back, but it was largely bacterial. Now we've done in recent years some antifungal antibiotics as well. That happens to be an antifungal antibiotic. And here's Cambridge, of course, the River Cam. I put that up because we got lots of our phages that we've ever worked with from there. And these structures are weird structures. They're called gas vesicles, and I'll tell you a bit about that. And the other thing here is a stream in New Jersey, and where one of the bacteria that we work with came from. So I'll mention various things as we go along. So we worked on various bacteria particularly, but we have kind of favorite bacteria. One is Erwinia, now called Pectobacterium, and the other is Serratia. And you see the list here. That's a plant pathogen, causes soft rot disease and blackleg of potato. Some strains make a carbapenem antibiotic. Sequences are known now for many strains, and Erwinia has all the main protein secretion pathways, type 1 to type 6. And in fact, we discovered type 1, 2, 3, and 4 in that particular bug when we were doing a lot of work on protein secretion mechanisms. We've got good genetic systems, and now it's been renamed Pectobacterium and also Dicea. So those are the two names now. I'll probably switch from Erwinia back into Pectobacterium as we go along. Serratia are opportunistic human insect and plant pathogens, significant cause of multi-drug resistance in nosocomial infections. Some strains make prodigiosins, not every strain. And these are red antibiotics, which I'll say something about. And this particular strain, which came out of New Jersey by the Squibb Chemical Company, also makes a carbapenem antibiotic. So the ability to make a carbapenem is kind of a common theme here, and I'll be talking about that. Sequences are known for multi-strains now. There are soft rot assays and Caenorhabditis elegans assays for virulence available, and some good genetic systems for some strains. Okay, so here's the disease. Everybody's probably seen this in the domestic kitchen. Blackleg and soft rot. Blackleg is a disease in the field, and you can see these stems here are infected, and probably everybody's seen the potato rot, soft rot in domestic kitchen. So early on in that career, along with Jay Hinton, who's here, is a professor in Liverpool now, we were starting to do what you might call classical bacterial genetics at the time. And that system is basically you test, you mutagenize your culture, and then you test it on a host, in this case, a potato plant. And I hope you can see here, there's rotting on one side, that's the wild type, and then you can see some mutants that don't make as much rot. So we've been able to use that classical bacterial genetics strategy to identify genes encoding virulence factors. And you might expect, if it's a plant, there's a whole variety of pectinolytic enzymes and cellulases and some others as well, which are important in degradation of plant cell walls. So all of that work was done in the early days. I said that we were interested in protein secretion. So here are 6 types of secretion mechanisms, but type 2 is particularly was particularly interesting to us because a 2-step system, going across the inner membrane into the periplasm and then engaging with the type 2 apparatus to go through into this, to be secreted to the extracellular environment. We've done work on some of these other systems as well, but particularly type 2 was really important for the definition of protein secretion systems. I'll come back to that later when I talk about the regulation. Okay, so I don't have time to talk about all the different antibiotics and that we've worked with over the years. I'll focus largely on carbapenems and prodigiosin derivatives, prodigenines. We have worked on all these other ones, and this one at the bottom is the most recent one that we worked with, that we called solanamycin, and that's an antifungal. It makes an interesting molecule with interesting biochemistry. I'm not going to talk about that, but I'm going to focus on carbapenems and prodigenines. So why would anybody be interested in carbapenems? Well, I kind of got into this in a collaboration initially with Barry Bycroft in Nottingham and Paul Williams and the late Gordon Stewart. Carbapenems are extremely interesting antibiotics. They were discovered in the 1970s. Thianomycin was the first one. They're broad-spectrum beta-lactams. They're resistant to at that time they were thought to be resistant to the clinically encountered beta-lactamases, so that made them really important. They were effective at low dosage, but they were made at that time in low titer and complex derivatives were made. So we got interested in actually going after the genes that made carbapenem antibiotics, and we did this with Barry and Paul and Gordon. They're not made by industrial fermentation, at least they weren't then, but they're made by chemistry, chemical synthesis, and the first one of these was imipenem, which is a really important carbapenem. Okay, so here are the molecules. Up here on the right is thianomycin, made by Streptomyces cattleya. And here is the 1-carbapen 2-M3 carboxylic acid that's made by the Erwinia strain that we've worked with. Indeed, it's also made by that serraceous strain. So this simple, the most simple, most basic carbapenem, both those 2 bugs make that. And because of the genetics, we started off trying to tackle the biosynthesis of that because of the use of bacterial genetics. Okay, so we started to get some mutants, and this, again, back to standard bacterial genetics. We isolated mutants that were defective in the production of carbapenems, and here's just an old plate that's showing you the production of the antibiotic shown here around the colony. Here's the wild type down here, and here are 2 mutants. All these ones on the side are mutants of different classes, but these 2 here are starting to make the antibiotic because these are cross-feeding assays, and the mutants themselves are in here and around the side. They are carbapenem negative. But the noticeable thing here is that the ones where you do a cross-feeding, class 1 and class 2, put them together, you restore antibiotic production. And at the time, we thought this is just classical centrifism. But actually, it turned out to be a lot more interesting than that, because those guys that don't make the antibiotic are also phenotypically Rex minus. That means they're defective in exoenzyme production as well. So they're highly pleiotropic. So the question is, what's going on in those strains? And the reason is it's not a syntrophism in a kind of classical sense. It's because they're one class 2 mutants don't make oxohexanyl homoserine lactone. That's the molecule. Now, that molecule was really important because if you chemically make that, synthesize it, and feed it back to a class 2 mutant, carbapenem antibiotic synthesis comes back, and exoenzyme virulence factor comes back as well. So they're highly pleiotropic, entirely due to that single molecule. And that molecule is responsible for bioluminescence, or the control of bioluminescence in Vibrio fischeri. So that was a curious connection. So this is what happens in the lux, or bioluminescence operon in Vibrio fischeri. This is a crude depiction of it, but there's both the biosynthetic genes and the autoinducer synthase, that oxohexanyl homoserine lactone gene, LUXI, in that same cluster. So essentially, that molecule is diffusible, and it's acting as a repressor, an activator, of the whole cluster. So ability to make light, bioluminescence, is controlled by the oxohexanyl homoserine lactone. And that happens to be the first of many different homoserine lactone derivatives which are capable of autoinduction. So up here I've just put a list of the kinds of phenotypes that people have associated with these N-acylated homoserine lactones, and the bacteria themselves which make those molecules. And in a good microbiology audience, you will instantly recognize that they're all gram-negative. Everything there is gram-negative, and there are plant pathogens, there's human pathogens, animal pathogens, and things that are nothing to do with pathogenicity. But everything in there is making an N-acylated homoserine lactone. And look at the diversity of phenotypes is quite remarkable. And just highlight this one. Type 1 and type 2 secretion is also controlled by that. So here's a bug, in the case of the plant pathogen, where the synthesis, the biosynthesis of the virulence determinants is controlled by N-acylated homoserine lactone, and the ability to actively secrete those, the construction of the secretory pathway is controlled by, in type 2 secretion at least, but also in type 1, by those kinds of molecules. So they're pretty important. And I'm sure everybody in this audience knows this is now called quorum sensing. So here's the basic. I'm not putting up any structures to show the modular nature of the LUXR type proteins. The LUXR proteins are the ones that control lux and the bioluminescence operon. But there are homologs of these in all these other systems that are encountered. So essentially you have a left and binding domain here on one side, and then the other one is a DNA binding domain. So you put those two together in a very primitive structure, essentially, where you're controlling the production of a terminal product, the target genes, via the signaling molecule itself. And that's a common phenomenon. Right, so here is the carbapenem production genes. It's the cluster that we've found. CAR A, B, C, a complete cluster. F and G are important for intrinsic resistance, but most of the biosynthesis is achieved by CAR A, B, and C, and a bit of these guys as well. So, the signaling molecule OHHL is going to bind to the CAR R, which is a LUXR homolog, the LUXR homolog protein, and when it binds back in this intergenic region, and that causes transcriptional activation of the cluster. Right. Okay, so the molecule is then ultimately made from that small cluster. And the biosynthetic route has been worked out. We didn't actually do this. It was colleagues in America that worked out the biochemistry. But those are, I don't have time to go into it, but in terms of the chemistry, those are some interesting enzymes involved here. Right, so of course I said that the ability to make oxohexanyl homoserine lactone was controlled both antibiotic production but also exoenzyme elaboration. So you won't be surprised to know that there is another Luxar homologue called VirR, which is important for the control for switching on all these enzymes, pectinase, cellulase, protease, etc. But they don't operate in exactly the same way. It's really a kind of largely a de-repression rather than a positive activation in the CAR-A system, the CAR-R system. So that's positive activation to switch on the CAR-R protein in the presence of the ligand, switches everything on for carbapenem. But virulence is controlled by the oxohexanyl homoserine lactone, the same signaling molecule, but it's actually by a de-repression system involving the enzymes involved there. Right, so the other bug that we started to work with is Serratia, a Serratia strain from the American Type Culture Collection, ATCC 39006. And it makes two interesting antibiotics. It makes the identical carbapenem as made by Erwinia, but it also makes this beautiful red pigment called prodigiosin. So the slight difference here is that it's a slightly different N-acylated homoserine lactone that is the main player in the Serratia strain. So it's BHL, or butanol homoserine lactone, in the Serratia, as opposed to oxohexanyl homoserine lactone in the case of the Erwinia. And there's the structure of prodigiosin and of the carbapenem. So it's exactly the same molecule, the antibiotic, in both the two bacteria. So this is just a depiction of the cross-feeding assay. So what we have here on the top of the plate is a SMA-I mutant. So that's the equivalent gene of a CAR-I. So the gene that's responsible for switching on production of the signaling molecule. And you can see that the CAR-I mutant is white, and that's because it needs to switch on the antibiotic. It needs to be cross-fed here from the wild type. So that's a pretty depiction of what's going on here. And it's a similar kind of cross-feeding thing involved in switching on antibiotic production. So we have the carbapenem antibiotic, we have the prodigiosin pigment, and we have two different molecules can be made, BHL and HHL, that can be involved in switching on the antibiotic and prodigiosin. Okay, so quorum sensing in Serratia acts by de-repressing the CAR-R gene. CAR-R gene gets switched on by the BHL, and then that happens to that doesn't require, if it's made, the protein, it doesn't require another ligand to switch the whole thing on. So once the CAR-R gene is de-repressed, and the protein then binds back in the intergenic region and switches on carbapenem antibiotic. So that's a slight tweak to the story. Now, it turns out that it isn't just Erwinia and the Serratia strain, but there's a strain of Phytopathogenesis that a group in the Pasteur Institute worked with that makes the same antibiotic. So that's 3 different antibiotics. They're made by Erwinia, Serratia, and Phytopathogenesis, but they're modified. They're regulated in slightly different ways. So as I said, In Erwinia, it operates by OHHL-dependent activation of the CAR-R protein. In the case of Serratia, it's BHL-dependent de-repression of transcription of the CAR-R gene. And in Phytopathogenesis, you can see from the diagram there, it doesn't have a CAR-R homologue, but it still makes the antibiotic, and that's because it's controlled by a different molecule. It's called AI2 dependent repression. And you notice also there's another locus that has some of those genes in Phytopathogenesis. So 3 bugs making identical antibiotic, but regulated in 3 different ways. Right, so I want to switch now a bit to talk about the prodigiosin. It's a complicated biosynthetic pathway. I'm not going to go through the actual chemistry of biosynthesis, but I put this up to make the point that there's a lot of genes involved in controlling prodigiosin biosynthesis. You know, here's SMA-I, SMA-R, the LuxR type protein, and various other genes involved in regulation, ultimately to make prodigiosin. Now, this is a rather bizarre situation because there's 14 biosynthetic genes involved in making that orange or red molecule. And there's more than 14, 15, probably more now, regulatory genes. So, you know, this bug puts a lot of effort into making and controlling the biosynthesis of that. And for some years, we wondered why does it, you know, do this? It does, you know, quorum sensing, it controls temperature, it's pH regulated, osmolarity, inorganic phosphate, just about everything controls prodigiosin biogenesis. So we kind of stumbled upon some story here. You see from the diagram here that you can isolate some pretty mutants, you know, things that are wild type or hyper producers, things that are kind of white, and things that are orange. And we started looking at some of these white mutants, and we turned up something quite interesting. This white, opaque colony phenotype is under quorum sense and control as well, but it's a different kind of quorum sense and control. Easy to isolate mutants, you can tell from the phenotypes. Facile mutagenesis screen is available for finding these white pigmented mutants. And initially we thought this must be some other secondary metabolite that makes this white pigment. Turned out not to be that. So here was an agar plate with the PIG-A mutant, the PIG-A plus, the PIG-A with a SMA-I mutation, so it doesn't make the signaling molecule, but you can restore the whiteness of this by simply adding back BHL. So that's why we thought, well, this must be another secondary metabolite. Turns out to be more interesting than that. And remember, this is kind of pre-ultra genomics. It turns out all the mutations that we got that produced this white pigment phenotype mapped to this 2-operon cluster. And it turned out, for our big surprise, that that 2-operon cluster is involved in making gas vesicles. Right? Remember, this is an enteric, a gram-negative. It's making gas vesicles, intracellular gas vesicles, which was quite a remarkable observation. So what are gas vesicles? Well, people who work on cyanobacteria and other bugs like that will be aware of that, but of course we wouldn't expect it to be made in a gram-negative enteric. So that was a big surprise. And it's affected by the light intensity and oxygen availability. So we checked this out. It turns out that the bug, the wild-type bug, is perfectly capable of making intracellular gas vesicles, and you can prove that by seeing them in that microscope, but you can also add some pressure to a bacterial culture, and that completely deflates the gas vesicles. So it's controlled by pressure. And if you try hard enough, you can see examples like this: a single bacterium absolutely packed with gas vesicles. Okay. So here's an example of a culture, a wild-type culture, grown for different time. Here we have a wild-type culture grown for 48 hours, and you start to see this appearance right on the interface. And the reason for that is that that's where the gas vesicles are accumulating. You get flotation. So that bacterial culture is actually depicting flotation of bacteria up to the air-liquid interface. And they don't make it when you don't have a signaling molecule, or whether you make it defective, or whether you put it under pressure. So here's a good example of it. Here's the SMA-I mutant. So it doesn't make the signaling molecule. And you can see that that interface here, the bacteria are essentially falling down. They're not total flotation. But you can restore everything by adding butanol homoserine lactone, and you can see that that will go right up to the interface. So, quorum sensing is control of prodigiosin, it's controlling carbapenem, it's also controlling gas vesicle biogenesis. And of course, because these are intracellular structures, by definition, that molecule isn't just a quorum sensing molecule, it's a morphogen, it's controlling the intracellular biosynthesis of a structure. So, why is this operating? Remember, this came out of essentially a swamp in New Jersey from the Squibb Chemical Company. And you start to ask questions, well, why would you want to float? What was the point in floating? Particularly in this case, because that bacterium is motile, it can swim. So why do you need to have flotation as a way of controlling this? So here's the idea. Essentially, you've got bioconvection. You can have these bacteria convecting themselves for a while, and maybe there's a kind of developmental switch happening where you shift from flagellar motility into a gas vesicle choice. And the thing is, if you look at these gas vesicles, because they float, they all go to that interface, and you don't need flagella or anything like that, because you can just float, right? So essentially, cell density is controlling the biogenesis of the flotation process. And you can clone this and switch it into E. coli and so on. It always works. Right. So what's happening here? Well, one idea is that maybe it's a kind of gang culture. You can starve competitors of oxygen, kill competitors and predators with antimicrobials, and defend your niche. So in a crude sense, what's happening here is oxygen gets burned up by the quorum sensing mediated culture that is in there. And because of oxygen dependence is a big factor in liberation, that controls carbapenem biogenesis, which you already know. But also these guys, if you're in an air-liquid interface, they're very susceptible to protozoan predators and things like that. So, it's a good idea to make beta-lactam, carbapenem, and also to make prodigiosin, which will attack protozoan predators. So that's where that culture came from, Cheesequake in New Jersey. It's the original home sweet home of 39006 before it defected to the east. And this is just a chemical summary. You keep talking the chemical language of survival. You've got the prodigiosin, you've got the antibiotics, and you make vesicles, you float, and through the effects of the morphogen, and like all things in microbiology, you know, you do whatever floats your boat. You do whatever is appropriate, and that's what these guys have evolved to do. Right, I want to switch now to talk a bit about bacteriophages. And in the blue there, you'll see bacteria have evolved many ways of avoiding lethality. Bacteria are susceptible to phage infection, but bacteria have evolved to get around the problem of virus infection. So that's the question. How do you defend the bacterial gang from viral attack? Now, of course, we all now know that there's a plethora of different ways in which that can be done. But when we started working on this, we worked on a process of a particular kind of survival against viral infection. So here's the, you know, if you're working on phage replication, you think about ways in which the bacteria can survive infection, and they're all depicted here. You can affect adsorption so that the phage doesn't get in, or you can control injection. But anything from here round to here, the end here, essentially everything in the physiological pathway, you could interfere in that process and block infection. So that includes obviously restriction and modification, and also includes CRISPR-Cas. So phenomenologically, that whole process is called abortive infection for obvious reasons. So the notion about abortive infections, or ABI systems, is that the infected cell gets infected by a phage, but you block the replication cycle, and that obviously the cell is going to die, but you survive in terms of a culture. They survive, the survivors aren't killed by viral infection. So essentially, it's an infected cell commits an almost altruistic suicide. All the rest of the culture is protected. So we weren't working on this, but I had an undergraduate project student, Claire Kirkpatrick, and she gave her this project. There was a strain called Erwinia atraseptica, strain ECA 1039, which we got from Ian Toth's group in Dundee. And it turned out that there was a little cryptic plasmid in that strain. We weren't looking for this. It was a project student found that plasmid. And that's it there, 1039. And you'll see that there are, most of the rest of it's uninteresting, but there were two little genes in there, one called TOXI and one called TOXN. And TOXN, there was only one other protein in the database that had any homologue, and that was from a gram-positive Lactococcus lactis. So here was a situation where TOXN, or the ABI-Q as it was then, had the same ability to make this toxin. We didn't know what was this little thing upstream, but I'll tell you about that in a second. So that was a fortuitous, a serendipitous observation by an undergraduate project student. Okay, this is looking a bit cloudy, but basically the TOXN is depicted there. So essentially I've taken the plasmid and linearized it, and it turned out that it does indeed have an abortive infection capacity. So you see here, if you just take the tox-I, tox-N cluster, that bit, and you put those together, then against these two phages, A2 and M1, there's a 5 or 6 log reduction in ability to replicate. But if you put a frame shift mutation in the tox-N region, you essentially restore back to infection. So it is indeed an abortive infection capacity. This is a more linearized, so here's the tox-N, but upstream, that little thing that we thought was a gene, there's 5.5 sequence repeats there. So it's 5.5 repeats, and then the toxin protein. And initially we thought that there might be an open reading frame, but we threw that out. So I won't go through this in detail, but the point is here, if you express the tox-N, basically everything gets killed. And if you've got the I gene beside it, the bacteria will be able to replicate. So what's going on there? Well, at this point, there were 2 types, I think, if I remember correctly, type 1 and type 2 toxin-antitoxin systems in the literature. And one was a full-length mRNA system, and the other was a protein-protein system. So it turned out that what we had discovered is this third type, type 3. And this is a combination of the toxin, the protein that gets made, and these antitoxic repeats that are immediately upstream. And there's a stem loop in between here. So that became interesting, and in collaboration with colleagues in biochemistry, we got the structure, and it turns out to be this beautiful structure. It's a novel type at that time, type 3 protein RNA bifunctional toxin-antitoxin system, and a phage abortive infection system. So we have these repeats of these kind of hanging together one by one. And apart from the aesthetic beauty of this, it's quite remarkable that this should be found. And it was found fortuitously in a cryptic plasmid in a plant pathogen. So if we look at the tox-I structures, their pseudoknots are formed in and pseudoknots are interesting structures if you're a structural biologist. And I won't go through all of this, but the tox-I itself, these repeats, form RNA pseudoknots. If you look at the genomes of lots of different bugs, you can find that these kinds of structures can be made. So for example, you can find a tox-N homologue in lots of different systems here, including gram-positives and gram-negatives. And then there are these repeat structures, which are all different, but you can make different structures that essentially probably bind to the toxin in different ways. So if you look at the structure, I showed you the first one, which is from the Serratia, the Erwinia case. But there was another one that we found from a gram-positive, from Bacillus thuringiensis. And you can see you don't need to be a structural biologist to realize these things are remarkably similar. And we thought that would mean that most of this would be repeated in different systems. But then we got the sequence of this protein called captin, CPTIN, from Eubacterium rectali, and you can see that looks completely different. And it's got longer repeat elements that are kind of binding these together for for the protein, which is a toxin type protein, but there's only 2 in this system, whereas there's 3 in these others. So, there's different ways of doing this. And also, I should say that the toxin-antitoxin relationship is specific. There's no functional mix and match with these, the gram-positive and the gram-negative versions. So the question is the evolution of the quaternary structures possibly driven by the pseudonautid RNA length? Okay, so the model is that you would have, here's the toxi and toxin, the repeat elements, the protein itself, toxin, and that has, it's an RNase, so it's capable of chewing up the toxin, the toxi structure to make the whole package together. So you can kind of hypothesize about what possibilities might be achieved here. So you might have a situation where, say, the unknown phage product might activate the toxin, or you could release the free N toxin degrades cellular RNAs as well as itself. And the most interesting thing is how might certain phages be able to escape this system? Remember, it's an abortive infection system. How could a phage escape the system? And here's a possibility here. You could have a mutant phage product that does not activate the toxin. That's a possibility. Or you could have a phage that produces an alternative toxin inhibitor, allowing replication to go on. So it turns out that's really interesting. What are the identities of the escape phage products? And why is it that some phages are extremely susceptible to this and some just ignore it? So this is work that was done by Tim Blower. This is a phage that we had for some time. It was isolated by Terry Evans, I think, by TE. And it's a generalized transducing phage, so that was why we started working on it. But it turned out when Tim analyzed this, you could get escapes spontaneously of this TE phage, but at remarkably low levels, 10 to the minus 8. So these are rare, very rare escapes. And when that was examined in some detail, the escape phage of TE expands the size of a very specific genomic locus. And once Tim pinpointed the nature of the locus, further examination showed a remarkable event that was going on. Because if you, I'm not showing you the sequence here, it's just the diagrammatic representation. So here is the wild type of the TE phage in that particular locus. And here is the whole toxi sequence, remember 5.5 repeats. But what turned up were a whole bunch of reiteration of these little sequences. And that's why they're capable of escaping. And this is a stunning result that Tim found, because these are extremely rare events. So he came up with a model for what we called pseudotoxi, and I won't go through that, but essentially those are versions of the real sequence that have evolved in these phages. So in other words, TE escapes abortive infection by an RNA-based molecular mimicry. Right? That's a stunning result. It's 10 to the minus 8 frequency, but as you see from the sequence, it's relatively easy to find these guys if you put that strong selection pressure on. Okay, so a kind of summary here is we have a toxi that makes an RNA, we have the toxin, and then those two kind of bind together, being infected by an enormous phage there by the look of it, and you make a viral product, and then you make the activated phage product, and you escape the tox-IN system by a couple of different mechanisms. So there's more than one way of escaping this. And that begs lots of questions about fitness impacts and evolutionary role, and is it early altruism and so on. Okay, so I just want to make the point here that I'm always reminded of Bruegel's painting of the Triumph of Death. It's a pretty horrific display here of people having their throats cut and taken out of coffins and then killed again. And I mean, it's just a horror show, hence the name, The Triumph of Death. It's a bit like a BBSRC committee, but I think so you could adapt or die, you can or embrace the inevitable, but you might actually adapt by dying. So that's basically the implication of what could happen as a way of escaping this system. Because if you've got lots of bugs, one that's been infected just needs to die by this system for the rest to be saved. And I like to put this up because I know this is a different society, but I used to be a member of the British Society of Plant Pathology, and this is this lovely little motif marker, which is remarkably similar to what biology has invented for us. So it's the answer to why work on phages, work on a cryptic plasmid from a plant pathogen, Erwinia or Pectibacterium, is interesting from the perspective of virus-host interactions, ecology, where the bug came from, the physiology process, structural biology, obviously, and protein-RNA interactions, and even perhaps the evolution of altruism. So, it's pretty easy to defend working on phages, I think. Maybe there is intelligent design, right, if you look at that structure. Okay, I'm going towards the end now. I just want to, say something about all the people that have been in my group over the years who've done this work. And I've mentioned Jay before, but Jay was my first PhD student. This was a master's student, Stella, in Canterbury. And Jay was my first PhD student who actually moved from Canterbury initially to Edinburgh, but then to Warwick. And you'll recognize, I'm sure, lots of these names here. I won't go through all of them. But essentially, the first two columns there, they were the Warwick era. And from then on in, it's the Cambridge era as new people came in. But there are lots of people, I'm sure everybody recognizes some of these names, including there's a couple of former Fleming winners in this list. Peter Finneran and where is she? Must be on the next one. And that's this is a bunch up to the most recent 94 who are achieved lots of different things. So as Churchill said, this success consists of going from failure to failure without the loss of enthusiasm. I always used to tell students that this is, you need to hang on in there because something good is going to happen. And then Marjory Stephenson Prize winners, the nominators, Tim Blower, who's in Durham but is now defected to Boston, and Sarah Coulthurst from Dundee. These are former PhD students and postdocs who have made a major contribution in all of those areas. So this is the group that moved from Warwick in 1996 to Cambridge, and that's, of course, the Hopkins building that they're going into, and that's the building that Marjory Stephenson worked in. And I have no doubt that you recognize a good few of these. There's 1, 2, 3 I can see instantly who went to the Sanger Centre after that. And then this shows a list of people who are master's students, PhD students, and postdocs in there. And that's the most recent, 2019, in a group meeting that we had in Wolfson College. And I was just looking at these people, postdocs and PhD students. I can't remember exactly, but I think there was about 12 or 13 of them were from all different countries. I mean, quite remarkable. They were from Chile, from France. I mean, there's a whole list of different countries who were coming from China, to Korea. It's an interesting bunch of people. Of course, there were external collaborators as well. I mean, obviously people who were in the biochemistry department or elsewhere in Cambridge, in chemistry, in plant sciences, and in the Department of Applied Maths and Theoretical Physics, the flotation story, Ray Goldstein. But the bacterial things in pathogenesis was Ian Toth from the James Hutton Institute. And Ian was an early PhD student as well. Now he's a professor up there. And I mentioned Barry Bycroft and Paul Williams and the late Gordon Stewart, who we all interacted together in the carbapenem and quorum sensing story. And we'll recognize this guy down here, Gordon Duguid. I don't know where he's from, but I think. And Nick Thompson, and Del Pickard, who I saw earlier today. Del was interesting because we had an interaction where Del was able to provide lots of what we call Viuna-like viruses, which I don't have time to talk about, but that's a really interesting story as well. Okay, so I want to finish with this story because many of you will know I like to use this poem by Hugh McDermott. And there's a quotation there from Sir Ronald Ross. But I used to, when I was teaching in biochemistry, I always used to use this to tell students, that top line there. Without some chemistry, one is bound to remain forever a dumbfounded savage in the face of vital reactions. The beautiful relations shown only by biochemistry replace a stupefied sense of wonder with something more wonderful because natural and understandable. Nature is more wonderful when it is at least partly understood. And if you jump down here, you will begin to comprehend a little the subtlety and beauty of the action of enzymes, viruses, and bacteriophages, these substances which are on the borderline between the living and the non-living. So this is an important poem called Poetry and Science, made by Hugh McDermott. And I think, the point is, as you see this line here, therefore I seek a poetry of facts. And he's contesting the notion that some people would push that, if you get interested in all this kind of detailed stuff, you're losing out on the beauty and the subtlety. He's making the point. It's anything but. It's exactly the opposite. So that's Marjory, and the seminar room is still there in that building, and in Hopkins Building in Tennis Court Road, and I think she made a tremendous contribution to this society, but also in chemical biology. It used to be called chemical biology instead of biochemistry, but chemical biology. And it's nice to see the wee dog there, Judith. I told my ex-postdoctorate I couldn't resist the notion of, at this point, saying, they're killing the dogs. So I think that's enough.